Hello, my name is Katharina Waury. I am a PhD candidate at the Vrije Universiteit in Amsterdam and an early stage researcher within the Myriad project, which focuses on biomarker research for dementia. And today I will present our work on prediction and analysis of extracellular vesicle-associated proteins. I will start with some background on extracellular vesicles, or short EVs. I will explain their function and their potential within biomarker research. Generally, EVs are membranous structures that have been found to be secreted by virtually all cell types and organisms and can contain many different molecules such as nucleotides, lipids, and metabolites, but in this project we focused strictly on protein cargo of those vesicles. EVs can be classified based on differences in size and biogenesis. Exosomes are the smallest type of EVs, with a diameter lying between 30 and 150 nanometers. They are formed by inward budding of endosomal membranes, and this creates so-called multivesicular bodies. Those can afterwards merge with the cell membrane to release those exosomes into the extracellular space. Microvesicles range in size between 100 and 1000 nanometers and are generated directly at the cell's plasma membrane by outward budding. Apoptotic bodies are secreted by dying cells and can have very differing diameters between 50 and 5000 nanometers. We focus here only on the exosomes and the microvesicles. These types of vesicles have previously been thought to be a way of cells to cleanse themselves of unwanted material, but nowadays their main function is thought to be cell-to-cell communication and cargo transport. EVs have also been associated with disease, especially cancer and neurodegenerative diseases. Their involvement in brain disorders is not yet fully clear though, and quite ambivalent, as both pathogenic and protective roles of EVs have been suggested. EVs might be the way of a cell to clear out aggregating proteins that are a hallmark of several different dementias, which might allow the cell to survive longer. It has also been proposed that EVs are a way to transport necessary protective proteins, such as heat shock proteins, to cells that are already impeded by those aggregating proteins. On the other hand, EVs might be involved themselves in the propagation of neurodegeneration by transporting aggregated proteins to yet unaffected cells, and thereby spreading actively the pathogenesis. Because of this quite complex picture of the association of EVs with disease, medical research has also increasingly focused on them. Especially for biomarker research, EVs are very interesting. For one, they are thought to be a good indicator of the processes occurring in the donor cell, and secondly, they are often easily available as they can be found in body fluids such as cerebrospinal fluid or blood plasma. Those EVs and their cargo might give insights into what is happening in the tissue without the need to directly access it, as for the brain that is very difficult to achieve in living patients. We were therefore interested to understand what affects protein-EV association. While EVs are a biomarker source, the proteins contained inside are not directly accessible, and specific methods for the isolation of the vesicles and the detection of their cargo is required. The prediction of protein-EV association could therefore help inform biomarker research and improve that process, and we wanted to establish if that prediction is possible, especially if only the protein sequence is known. I will now explain the workflow that we followed to generate our datasets and to produce the classification model for EV association of human proteins. We started by accessing EV databases, Vesiclepedia and Exocorder, that collect identified cargo from EV studies. This created a dataset of 13,648 human proteins that have previously been found in EVs. As we also wanted to use a negative dataset, so proteins that have never been found in those vesicles, we used a recent version of the entire human proteome from the UniProt database. The idea was then to assign the human proteins not found in the EV dataset as non-EV proteins. For the set of 20,385 proteins, we first generated features. Sequence-based features include composition of amino acids, molecular weight, Isoelectric point, and instability, and we also use predictions from net P for secondary structure, disorder, and surface accessibility prediction. Created annotations included mostly post-translational modification annotations from databases such as UniProt, Phosphosite Plus, and IPTM. Both created datasets were then filtered. We chose to exclude any proteins from the study that have only been found in 1 or 2 EV studies, as we considered those ambivalent. The EV protein dataset therefore only contains proteins that have been found in at least 3 different experiments. The human protein dataset had to be filtered for only mass spectrometry detectable proteins. The reason for that is that early on in our project we noticed that EV proteins were strongly positively correlated with size-based features such as molecular weight or sequence length. As the vast majority of proteins in the EV databases have been detected by mass spectrometry, we realized that this might indicate that our data is biased towards proteins that are detectable by MS, and thus towards proteins with a higher molecular weight, as those are more easily detectable by mass spectrometry. To remove this bias, we excluded the human proteins that have not been detected by MS experiments in general. In the plot here, you can see the effect of this filtering step on the distribution of the molecular weight on the datasets of all proteins, the EV and the non-EV proteins. The distribution of non-EV proteins in red is affected the most by the filtering step, as many low molecular weight proteins were deemed non-EV, but are actually simply not detectable by mass spectrometry. You can also see that after filtering, the distribution of molecular weight is much more similar between the 3 groups, although the EV proteins still show a slight shift towards higher molecular weight. We then, after those filtering steps, created 3 datasets. For all EVs, and specifically for exosome and microvesicle associated proteins, as we were interested to see if EV type specific prediction shows differences to general EV prediction. For all 3 datasets, we built 2 random forest classifiers. One classifier using all features that we generated and one using only the sequence-based features. We then looked at which features were most important for classification to understand the properties better that affect EV association. Our aim was this to answer 2 questions. For one, can we predict EV association? And if yes, how and why can we predict it? To answer the first question, if we can predict vesicle association, we looked at the performance of our classifiers. To the left, you can see the ROC AUC curve for general EV association prediction, and you can see that the random forest classifier achieved an AUC of 0.82 when all features were used, and it's dropping a little bit to an AUC of 0.75 when only sequence-based features are taken into account. You can see a similar pattern also for the exosome and microvesicle specific predictor, and you can also see that the performance of those is slightly better than for general EV association prediction. To answer the question how and why we can predict EV association, we performed a feature analysis, meaning we looked at which features were most important for the correct classification and which features were most positively and negatively correlated with each respective EV class. Here in the table, you can see the top 10 most positively correlated features for extracellular vesicles, exosomes, and microvesicles. And from those, we try to derive general properties of EV proteins. One thing that becomes clear quite fast is that post-translational modifications are strongly positively correlated with EV proteins. As you can see, in all 3 of our datasets, the 5 top most positively correlated features are PTMs. From this, we conclude that one of the properties of EV proteins is that they are rich in PTMs. To give you an even better idea of how much in EV proteins PTMs are enriched, we plotted the fraction of PTM annotated proteins both in the EV and non-EV group. And as you can see in the blue bars, for every one of the modification types that we looked at, that there is a clear enrichment in the EV group compared to the non-EV group, especially for palmitoylation and acetylation. But you can also see, looking at the yellow bars, which show the results from post-translational modification prediction, that we could not replicate those results with the predicted PTMs, as Often the relative difference between the two groups is lost, such as with palmitoylation, and also we see a vast difference between the absolute numbers of annotations, such as in ubiquitination and glycosylation, where there is stark differences between annotation and prediction. Another feature that we saw strongly positively correlated is the fraction of exposed aspartic acid, a charged amino acid, or of polar amino acids in general. From this, we conclude that another of the properties of EV proteins is that their surface is more polar than that of non-EV proteins. Now we are looking at the most negatively correlated features for EVs, or in another way, the features that are most correlated with the non-EV group. One thing that is immediately clear here is that often the features that give information about the fraction of hydrophobic amino acids are strongly negatively correlated. This is true for a general amount of exposed hydrophobic amino acids and specific ones such as leucine, tryptophan, and phenylalanine. And from this we conclude that EV proteins have lower hydrophobicity compared to non-EV proteins. Another property that we found to be negatively correlated is the isoelectric point. The isoelectric point usually shows a bimodal distribution, as every proteome can be divided into a set of more acidic and more basic proteins. As you can see in the plot in the lower right corner, we see this bimodal distribution both for the EV and the non-EV group. But the EV group shows an enrichment of proteins with a lower isoelectric point and therefore an enrichment of more acidic proteins. We also found that the instability index was a very strong feature for prediction, as well as a strongly negatively correlated feature. As an instability index higher than 40 indicates that a protein is unstable, a lower instability index for the EV group indicates that those proteins are more stable than the non-EV proteins. We now come to the main conclusions of this work. The first one is that while EV databases are a valuable resource, they are clearly biased towards proteins detectable by mass spectrometry and therefore towards protein with a higher molecular weight. It is essential to be aware of this bias in proteomics data and also to correct for it in similar studies that are mainly based on mass spectrometry data. We also showed that the prediction of EV association is a possible but not trivial task. It is possible as a simple random forest classifier achieved an AUC of 0.82 for EV classification and an even better performance for EV type-specific classification. It is, however, not trivial, as several different features had to be used to achieve this performance, and not one feature alone allowed the correct differentiation of EV and non-EV proteins. We also found clear differences in the physicochemical properties of EV proteins compared to the non-EV group. The more polar surface of EV proteins that we found can explain the observed negative charge of EVs themselves, as many proteins are also transmembrane or attached to the outside of the vesicles. The difference in the distribution of the isoelectric point, with EV proteins being the more acidic ones, could indicate the previous subcellular localization of the proteins that become EV cargo. That is because previous studies have shown that differences exist in the isoelectric point between the proteomes of cellular compartments, and the most acidic proteomes found belong to the cytoplasm and the lysosome. As the lysosomal pathway overlaps with the exosomal pathway in the beginning, an overlap of the proteomes is also understandable. PTM annotations were the most important feature that we found for correct EV classification, and the systematic analysis of the EV databases showed that those modifications are also clearly more abundant in the EV proteins. This supports the previous findings that PTM seem to be incredibly essential for the correct sorting of proteins into different types of EVs. Thank you for your attention, and I want to say a big thank you to our group at the VU, and especially to my colleagues Dea Gogishvili and Sana Aben, who I have been working with on this project, as well as the Myriad Consortium.